Cloning vectors, quick quiz, class 12 biotechnology.
Which of the following is an essential feature of a cloning vector?
Option A, origin of replication. Option B, lysozyme binding region. Option C, large genome size. Option D, heat shock protein coding region. The answer is option A, origin of replication. Question 2. What is the primary function of plasmids and bacteriophages in bacterial cell? Option A, to provide energy to the cell. Option B, to replicate independently of chromosomal DNA. Option C, to produce proteins. Option D, to degrade foreign DNA. And the answer is B, that is to replicate independently of chromosomal DNA. Question number 3. What is the role of original replication in a cloning vector? Option A, it helps in the selection of transformants. Option B, it controls the copy number of linked DNA. Option C, it provides antibiotic resistance. Option D, it facilitates ligation of foreign DNA. Answer 3 is B. It controls the copy number of linked DNA. Question number 4. Which of the following is considered a useful selectable marker for E. coli? And the options are A. Glucose, B. Ampicillin, C. Sodium chloride, and D. Lactose. And the answer is B. Ampicillin. Question number 5. What happens to the antibiotic resistant genes when a foreign DNA is inserted into a cloning vector? It becomes more effective, that is option A. Option B, it is inactivated. Option C, it is duplicated. Option D, it remains unchanged. The answer is option B, that it is inactivated.